Thank you for taking the time to learn more about the nCounter analysis system. The nCounter platform is an easy, quick way to generate high-quality expression or copy number data on large numbers of genes. 2/3 of our current customers work in cancer research, and they're our fastest-growing customer base. There are a number of reasons why our system is especially well-suited to oncology studies. Cancer research often focuses on particular biological pathways. Because the nCounter system can multiplex up to 800 targets, you can study multiple pathways in a single reaction. Our platform also does extremely well on formalin-fixed paraffin-embedded tumor samples, which are one of the great resources of the oncology community. These samples can be very challenging to work with in other assays. Deletions or amplifications of particular regions of genomic DNA are a hallmark of tumor tissue, and the nCounter copy number variation assay is a simple way to characterize these. All of the nCounter assays are sensitive, precise, and reproducible. They're also very easy to run. In the next few minutes, I'll go over the highlights of the technology and how it might apply to your research. The recent advances in genomic information are impacting the way we think about biology. Complicated multi-gene pathways are one of the emerging units of biological analysis. The focused study of particular pathways can lead to rapid insights into biological function. This slide shows typical gene numbers for a variety of pathways. You can see that they range from the low tens up to several hundred genes. There are a number of ways to study hundreds of genes at a time. Analog technologies such as microarrays and quantitative PCR don't offer the precision and the dynamic range that digital technologies such as next-generation sequencing do. Sequencing generates high-quality digital data, but it requires extensive sample preparation as well as bioinformatic resources. It can be cost and labor prohibitive to study even a moderate number of samples using this approach. Like sequencing, the nCounter system also generates high-quality digital data. It can analyze up to 800 targets in a single reaction. Unlike sequencing, nCounter has an extremely simple workflow and can be very time and cost-effective for large numbers of samples. Our customers are using nCounter to study multiple targeted pathways. They typically create a core gene expression code set of several hundred probes, which allows them to analyze half a dozen different pathways, as well as other specific genes relevant to their study. They complement these gene expression studies with analysis of the copy number variation in the genomic DNA of the tumor or a microRNA profile of the sample. As I mentioned, one of the huge advantages of the nCounter system for oncology research is its compatibility with FFPE samples. Formalin fixation and the subsequent processing of tissue samples modifies and degrades the nucleic acids. The analysis of both RNA and DNA from FFPE samples can be really challenging. nCounter eliminates this challenge. Our assays generate data that's on par with the data generated from matched fresh frozen material. The nCounter assay does well on FFPE samples for two main reasons that involve both the method of the assay and the length of the probes. The assay involves direct hybridization with no amplification to approximately 100 bases of target. This direct hybridization isn't impacted by modification to the nucleic acids, and the small target region means the assay can withstand significant amounts of degradation without a decrease in data quality. Our customers have analyzed thousands of FFPE specimens with nCounter, and many now consider it to be the gold standard method of analysis for these samples. Let me briefly walk you through how the nCounter system works. As I mentioned, we select a 100-base target for each gene or region of interest. We design a probe pair consisting of a 50-base capture probe and a 50-base reporter probe directly adjacent to each other. The capture probe is biotinylated, and the reporter probe carries a 6-spot fluorescent barcode. Each target is assigned a unique color combination. We can put up to 800 probe pairs in a single reaction. Following hybridization, we capture the target probe complex on the surface of a slide and stretch out the molecules so they're all oriented in one direction. We then use fluorescent microscopy and image analysis software to count the captured barcodes. This generates digital data, with each count being one molecule. The assay has very high precision and over 5 logs of dynamic range. Here's a close-up of the slide's surface to give you an idea of what the actual images look like. We aren't quantifying based on fluorescent intensity. We're generating a digital result by counting individual molecules. The data itself is simple. The software tabulates the number of times each barcode is counted and returns a list of genes and a corresponding number of counts for each. Now I'd like to mention 3 recent examples of our customers' research that each highlight a different aspect of the system. This paper by Patricia Rees et al. From the Ontario Cancer Institute demonstrates the advantage of nCounter in evaluating FFPE specimens. The authors compared data from paired fresh frozen and FFPE samples of oral carcinoma tissues using nCounter and quantitative qPCR. The figures on the left show nCounter data which had a Pearson correlation coefficient between FFPE and fresh frozen of 0.9. The figures on the right show equivalent data for qPCR, which had a correlation coefficient of 0.5. The authors conclude that nCounter is a better platform for gene expression analysis of FFPE samples. It's worth noting that this platform comparison study was undertaken completely independently by Dr. Reese and her colleagues. We only became aware of it at NanoString after it was published. In this next paper, Zifu Sun and colleagues at the Mayo Clinic used NGS to profile the transcriptomes of different breast cancer cell lines. They used quantitative PCR to validate the abundance of 3 relevant receptors, and then extended their validation by using NanoString to measure the abundance of an additional 236 cancer reference genes. The 4 small scatter plots on the left show the correlation between the RNA sequencing data and the NanoString data for 236 cancer reference genes in 4 representative samples. The large scatter plot on the right shows the log2 fold change correlation between the deep sequencing data and NanoString data for 25 genes that are differentially expressed between ER-positive and ER-negative samples. The very high degree of correlation, with an R value of 0.97, indicates the quantitative precision of the technology. Like next-gen sequencing, nCounter is a digital technology. Many of our research customers use nCounter as a rapid, cost-effective method of validating their RNA-seq results across large sets of samples. This third paper demonstrates another major advantage that I mentioned earlier, which is nCounter's reproducibility. Paul Northcott and colleagues at the Hospital for Sick Children in Toronto began by using nCounter to validate a 22-gene expression signature for medulloblastoma, and then expanded their analysis to include site-to-site reproducibility. To do this, they shipped 48 RNA samples to each of 2 additional sites, which performed the nCounter assay for them. The heat map show the expression levels of 22 genes across the 48 samples at each site. The scatter plot shows an extremely good correlation between the original site and each of the other 2 sites, with R-squareds of 0.97 and 0.98 respectively. The sensitivity, precision, and ease of use of the assay make the nCounter platform ideal for in vitro diagnostics. We're currently developing our first diagnostic test, a breast cancer intrinsic subtyping assay. The test is based on the PAM50 gene expression signature, which was originally developed by Chuck Peru and colleagues using DNA microarrays. The assay uses the expression profile of 50 genes to differentiate between 4 different subtypes of breast cancer. The positive results of our first clinical trial were announced at the San Antonio Breast Cancer Symposium last December. Our long-term vision is that there will be a diagnostic version of our nCounter analysis system in pathology and hospital laboratories worldwide. An important part of this vision is to provide researchers in the translational medicine setting with the opportunity to run their validation experiments on a platform that's excellent in the research context, and given the appropriate licensing and regulatory approval, can go all the way to the clinic. In summary, I'd like to highlight that the nCounter analysis system was developed to facilitate the study of focused gene sets with very high performance and relative ease. Oncology researchers make up NanoString's largest and fastest growing customer base because the nCounter system offers a number of benefits that are particularly valuable for cancer research. The ability to analyze 800 targets in a single reaction, compatibility with FFPE, high precision from digital data, and excellent reproducibility. We're very excited to see scientists using nCounter to accelerate their research. New nCounter publications are coming out every week, and many of these are in the area of oncology. A variety of these papers are referenced on our website, along with much more detailed information about other aspects of the nCounter system. Thank you for your interest in NanoString. For more information, please visit our website or contact us to discuss your research needs.